So I hope everyone's back. I see all cameras are on. That's fantastic. How was it? You want to pick your favorite reaction from the bottom here? I don't know. So I joined. I saw a lot of people were working. 7 sessions actually really happened with some concrete working on the document. So I'm super excited about this. I think in the interest of time, we thought that we don't need a 15-minute transition time because we did need the backup. Yes. So maybe we can briefly go through the groups, and then since it's only 7, I think we may be able to leave a few minutes early. But let's start with pre-analytics. Of course, the most important and the most relevant. Garbage in, garbage out fully applies, right? Amanda, can I call on you to provide sort of a few-minute summary what you discussed? And
so the analytics, I had one attendee join me who is kind of new to the discussion, Darren Trainer. Darren, thanks so much for participating. We did spend a lot of time kind of discussing the alliance, the goals, where pre-analytics. So I had to kind of go and take back the historical picture. We shared and we went over the executive summary, which I think is a very good and valid summary of where we're at today. We still remain, so there's 2 things I'm really looking for as a working group is consistent engagement. So who's willing to really sit at this working group and roll up their sleeves and put in some work. And if you couldn't attend my work group, but are one of those people, please let me know. And 2, what are we aiming for? What we have achieved is a clear understanding that we are not looking to reinvent the wheel here and guidelines on laboratory variation. There's lots of documentation on how to control to do that. And we'll put some of those resources into our working group webpage. But what we're really trying to do is bridge this from a digital perspective of what things you need to consider in your pre-analytics for digitization and the impact of that from an AI machine learning perspective. So it goes back to maybe the path of least resistance is to continue to work on education, but also looking at a paper versus guidelines, things of that nature. But again, I need authors. I need folks who are willing to really come here, help us with literature review, really thinking of this as to, can we demonstrate in already documented literature where we've seen this impact the variability and the output there so that we can really address the problem and make recommendations for how we solve it and why that's important from a regulatory science perspective. So a lot of continued discussion because again, we're still trying to find our anchors in the team. And so I really am looking for those anchors. I need support and help with getting the consistency of the team going. So anyone willing to roll up their sleeves and help me, I would greatly appreciate it. So thanks again to my contributors, the folks from the scanning team who came over. We had some good discussions too. So just thanks everybody for their support today. And Ula, I'll send you some of the deliverables for the website after this meeting.
Awesome. Yeah, we looked, thanks a lot, Amanda. We looked into some of the relevant papers for the pre-analytics website. And there is a lot of action in this field, not necessarily in this particular group right now or in the alliance yet, I guess. But the histo QC, some of the H&E quality and stain unification, or making things look more uniform to humans, even when you have digital images, even artifact correction, etc. So we found some fantastic papers. So what we were considering, and I wanted to throw that out as an idea, is to really reach out to the people who actually work on this field and ask them what components might be relevant from a regulatory sciences perspective, right? Because it is sort of a niche, but it's extremely important. I think most, anatomic pathology labs have their H&E stain and they say, oh, ours is great, but how that holds up in comparison between institutions, we don't really know. I think there's some CAP proficiency tests that get that data, but I don't know whether that would be available for example, for the pre-analytics group to really review that as part of, let's say, a publication. But those were the notes that I had from, you know, the discussions we had in preparation for this meeting. Other comments or questions for Amanda?
Yeah, I'll go ahead and throw something out. I was listening to some podcasts. It seems like digital pathology podcasts are kind of a growing entertainment option and news option. And one of them was Marilyn Bowie at DPA talking about the CAP program. And I thought it was quite interesting how they are asking people to submit slides twice a year and then the corresponding images, and then they give feedback on any pre-analytic issues that they can detect from those slides and images. And you know, they're a part of this group. Someone is here today. I think where did he go?
It's on the call. I saw him.
Yeah, I think he might be missing in action at the moment, but you know, they're friends. Perhaps we can figure out whether those slides and images have some sort of value in the pre-analytic type of project. And you know, when we think about some of the projects that we have going on at the FDA, some of the things about how the slides and the color changes and characteristics, and just to really nail that down is some of the things that we look at.
Thanks, Brandon. No, I'm familiar with it too. And I think that's an important piece. We want to make recommendations that, again, people should be checking these boxes, but twice a year, kind of, you know, maybe it's, you know, it depends on the specific markers you're looking at. You've got to think kind of bigger picture too. So it's really around, and also from a regulatory science perspective, you want to know device manufacturers are considering these variables, right? As they look to gathering and creating the innovation that we want to commercialize. So I think there's things that we want, and we want to continue to advocate and educate that the laboratories and the digital laboratories are doing, but then how do we translate that into what's being utilized to develop this innovation? But thank you, that's great feedback.
Amanda, I think also that, you know, the variability that you observe from the scanners, and we're talking a little bit with Brandon earlier, and how do you expect a specific algorithm to be able, you know, to do the same good job across everything? You know, even from a regulatory point of view, that's a big challenge.
And modeling the different color distributions so that you can model one imaging system and model another imaging system. So as an algorithm developer, you don't have to have data from every one of those imaging systems, but you can use the models to generalize the algorithms to be robust to those differences in color characteristics. And, you know, it's got a lot of value for everybody. The people that want some feedback on their pre-analytics, the algorithm develops, you know, can get some of that modeling of the color characteristics. And I think that's kind of, you know, is a win-win on both sides.
And resolution, I guess, Brandon, too. I mean, I don't know if all the scanners can scan at the same resolution, right? So, you know, that's also, you know, I mean, so there are a lot of features there that.
Talked about the scanners too. Mm-hmm.
Sorry, what was that, Amanda?
No, I was just saying it talks about how pre-analytics bridges into slide scanning also. And we had that conversation on the work group too, and just where we kind of create a handoff in a way to the hardware piece of it.
Yeah, absolutely. And not only that, what I was going to say is even the scanner to scanner variation and how the scanner is calibrated really matters. Like we had, just anecdotally, we did a trial on a site and there were some errors, and we realized that a lot of their slides were blurry. Their scanner wasn't calibrated right. And you know, that matters too if you want to scan and then do analysis. So putting some standards in there is really important.
I mean, we're asking some of these new hospitals and health providers to start building up their patient records or their databases, and to give them an opportunity to get regular feedback on their SWOT slide scanning quality is not only helps them, but it also gives the other side, the developers, a chance to see what kind of variability is coming out of these systems.
Yep.
Perfect
And then,
segue
Amanda,
to
what good
Go ahead,
question? What came
Esther,
out
go
of
ahead.
it, like with regards to prioritization? Is it more the staining that we have to look into, or more the scanner? Because I hear different things. Like you can also think about how for easy biomarkers, it's we know that how often that goes wrong and how often they have to be restained to
Yeah,
make the correct diagnosis, or is it more the scanner itself? Yeah.
I think there wasn't, and it, maybe that's a good point, Esther, is we should prioritize which variability, which variables we want to try to control first, and that is most impactful. So I think, you know, everyone, we see it across the board. And so that's where, again, this is a big animal to try to really to think about from a development perspective and with pre-analytics. I see, Stephen, I think you have your hand
Yes.
up.
Steve?
Yeah, so I was, you know, we just have to be careful when we think about, well, what are the analytic characteristics that are going to affect the model, because depending on the way that the model is built and trained, we don't actually know what those pre-analytical characteristics are. So while it's easy for us to think of staining variability, that's one aspect, or blurriness in an image, that's another aspect. But I mean, there might be tissue tears and all these other variables. And that's something that came up in the AI ML group, is the fact that so much of the dependence of the algorithm and success of the algorithm is dependent on all of those pre-analytics. And so defining what that feature space in that is relevant and applicable to your model is important, even if we can't necessarily state what all those variables are.
Does this fit into any real world data happenings that are going on?
I know Esther had posted this a while ago from the DPA, right?
Yeah, so we do, we try to include as much as possible with regards to those type of data to collect that. But we also know that it's going to be very difficult to make any conclusions, but at least you can start seeing trends, right? So it's because probably the data that we're collecting is then not statistically significant, but at least the sample size is big enough to see trending. So if there's anything that you think that we could include, let us know, and then we will look into it. But we also have to be pragmatic, and that we're not not designing a Christmas tree, that we're collecting too much, and then the study itself will become too complicated. But there's things that we can definitely think to include there, and also how to start standardizing the data that you collect. How do you code that?
Right. Segue over to Scott from slide scanning. Do you mind providing a brief summary?
Sure, no problem. So, Garrett, Garrett was new to the group and he joined today. And so what we wound up talking a lot about is the histology leading into slide scanning. And we just wound up also talking about artifacts from a histological point of view, but more importantly, artifacts within scanning. And what I find really fascinating in each time this group gets together is how the commonalities just seem to form. So I'm hearing a lot about this from pre-analytics, and I suspect it'll carry on. So we were talking, so to go over the paper, so we really had no substantial changes to what's already planned out for the white papers and for the deliverables, but we are, I'm going to carry on forward, is possibly adding in classifying scanner artifacts as something that wasn't included before, because talking about whether you scan the same area twice, or more importantly, if you missed a piece or missed an area of tissue or something like that. So, but I just find it very interesting that it's a common theme that's going through this group. in many ways. It's all zeroing in on the same things. Another point that we brought up, and this was after we joined into the pre-analytics group that we had been discussing, which was, is there a sweet spot when it's better to capture certain types of artifacts at the scanner and never pass those images forward into the system, eating up even more time and lowering efficiency, versus what is better to be caught by other types of algorithms downstream further. We didn't really get a chance to start diving into that deeply. We just kind of started in on that, and then the timer ran out on us. But overall, it was, I think, a very good, discussion, and I've got another deliverable to add into the white papers and the group's activities. So, but I will echo what Amanda also said is the activity. I've got a few people that are contributing, but I can always use more. I definitely need people for like writing definitions and stuff like that, and it's challenging keeping the whole thing rolling forward, but we will get there.
So some of the things that you just mentioned, especially sort of like, you capture feature X at the scanner level, and then you mentioned sort of without moving this forward, reminds me a lot of some of the fallback or, sort of QA steps with next-gen sequencing, right? When you don't have a high quality run, that will not even be pushed to the pipeline, or, I think some of those things sounded at least a bit similar, but I don't know. Comments, questions for Scott or the slide scanning group? All right, I had at the next point, I had Mike. I think he had to jump off, but he provided sort of a summary. I can briefly read this out. So there were a couple of minor changes to the executive summary. He will get that to Ulla, and then the standards group discussed a bit closer how the outputs of the scanner could be aligned with some of the standards, especially, probably one of the in relation to one of the upcoming webinars with DPA, PIcc, and DICOM. And it would be great to have that also as part of the panel discussion. At least that's what he wrote. Is someone else still around from the standards group? I don't know who was in there, but if not, let's move on to the truthing group. Brandon, Kate, I know.
Sure. Yes, we started attacking the content that for the web page, and one of the things that I started with is, truthing is a great anchor, but really I'm in this for the truthing and performance evaluation. So I think just to label it as truthing is kind of not enough for me. And I want to check here that whether there's too much overlap with the machine learning group, or whether the machine learning group is on different architectures and study designs for training and things like that. So I want to know if that is too much overlap with the machine learning group, or whether I can take that under the truthing group.
Which part do you mean? Just performance evaluation in general?
Really put performance, because really I'm in this business to evaluate the performance of algorithms. And many of you might be able to guess. And really that's why I'm doing a truthing project to collect a data set of validations from pathologists, annotations, is really so that I can build up the tools and methods for doing the pathologist to pathologist agreement. And I see that as the baseline for evaluating the algorithm to pathologist agreement, where the algorithm to pathologist agreement is kind of the performance of that algorithm. And those methods, I think there's plenty of room to innovate on the statistical methods there, and there's plenty of need, I think. especially when we're talking about truth from pathologists. So I just want to grab that space in the truthing, unless it's too much overlap with something else.
I think that's a great idea, especially, I mean, apart from the output in terms of like, you know, the performance during training and validation sets, I think there's distinct things. The agreement between pathologists is just, I think, the surface. If you start thinking about, we use histology to predict mutations, there is no pathologist agreement on that because pathologists can't do it, but machine learning can. At least, you know, the current literature suggests that. So the question is, you know, whether there is a statistical framework for how to report this or how to assess it, right? Because ultimately that's what I presume the regulator has to deal with anyway.
And I'll say you hit on the other expanding of our scope, if you will. You know, my project has been about pathologists, and that's really my niche currently at the agency, is about studies where clinicians evaluate images. But that's not the only way to get ground truth. And in fact, if I speak in like it is the only way, I get hammered by people that say there's other assays, there's patient outcomes. And so I wanted to at least put that in our executive summary, that truth is, and I see it, and I'd be happy to hear any other categories, but truth by pathologists, truth by other assays, and truth by patient outcomes are the big buckets that I put them in. And if you have any others, you can shoot them to me later or in the discussion. And so that kind of really drove us to rewrite some of the executive summary because it was a bit too much pathologist oriented as far as truthing.
I think the other way of wording that, other than performance evaluation, is it sounds more like the classification of ground truth has to be done somehow, right? It sounds like you have an internal rubric for what is the ground truth. Is it survival? Is it other outcomes? Is it subjective measures? Is it ICD codes, or is it just agreement between pathologists? I haven't seen a classification for that. I mean,
well,
everyone
I'd like
talks
to
about
stick
outcomes,
with performance because I want to be talking about the performance of an algorithm or the performance of an imaging system. And you know, we talked about the variability from clinicians or pathologists, but when you're talking about other assays, you know, especially for algorithms, those other assays might be from a section that is not the same as the one that the algorithm is working on. And so there's a little disconnect between the actual material being evaluated, or you might need to destroy the specimen that you had on an H&C slide in order to run it through another assay in some situations. And then patient outcomes, you know, you think of those as the gold, those are gold standards often. But when you're trying to grab it from the patient record or a registry, then we're talking about there's going to be noise in that reporting. There's going to be missing data in that reporting. So, but I think all of this still is how do we use that data to evaluate the performance of an algorithm or the performance of an imaging system. So I still, you know, I want to hold on to performance. And that's just how I think about the problem. But that's where, you know, other projects, our project that I have going right now is about pathologists establishing the ground truth. But I certainly think I will move ultimately to some others, or actually not me move, but I'd like to see other contributors be establishing methods and protocols or how or things related to these other ways to establish truth. And just for everybody's just to share, you know, the research I'm doing is about a quantitative biomarker, TILs in breast cancer. But I think the evolution of that work is going to take us to truthing segmentations and truthing grading scales. You know, those are very different data types and require different statistical methods and metrics. So those are some directions.
The ground truth in the TILS project is what, the pathologist count, or?
So it's a, well, imagine there is some truth. It has units, and it's the density of TILs in a stromal tumor area. And so that's quantitative. There is some kind of density, whether we can get it exactly or not, but it does exist in my mind. And we're trying to get at it from the pathologist. And then we, you know, ultimately we know that that's probably got a lot of variability associated with it. And, you know, essentially once we can pair this with the algorithms providing that measurement, we probably will get to a point where the algorithm is more trustworthy than and quantitative really is what I don't mean trustworthy, but quantitative at the granularity of percents, you know, singular percents, whereas, you know, the granularity of making a clinical management decision is you know, where we are right now and where we need to be for managing a patient. But if we really want to be pushing all this technology forward, we want to be quantitative, that is at the resolution of the observable, of the physical phenomena.
I think also is the consistency of rules, right? Because that's what the algorithm is giving you. It's giving you a consistency, you know, of rules, you know, where humans weigh different rules, different way when they observe something. So.
Right. And that's where getting to more qualitative scoring systems, you know, once we can at least establish that the algorithms can kind of fit what the humans are doing, then we can maybe get quantitative on those rather than relying on these kind of artificial rubrics for the Gleason scale or HER2 or all that stuff has some sort of physical observable underneath it. And if we can get to those quantitative biomarkers in a more quantitative way, and I think patient and public health would be improved.
Other comments or questions to the truthing group?
I do have an additional comment, which is, it sounds like your performance and evaluation interest is really separated into two areas, because there's the question of algorithmic performance evaluation, but then there is also, you said imaging system performance evaluation, but that could be essentially evaluating the performance of the pre-analytic steps of the imaging, of the scanning, et cetera. And it seems like that is a, I don't know, do you consider those the same sort of unified whole of performance evaluation, or do you really consider those two separate areas that could be sort of done in parallel?
So the pre-analytics really doesn't come up to me personally. When I think about the imaging system, I mean, I would say more, what is the image is taken for? Perform some task, some clinical task, and then that who is doing that clinical task? It's either going to be the algorithm in the future, or it's the clinician, the now. And so seeing how well, that's why pathologist agreement is as important as algorithm to pathologist agreement for my work and this as it relates to this work group. I don't know if that exactly answers it, but most times when I try and think of how would I evaluate an imaging system, there's the technical performance assessments stuff, which might have some pre-analytic stuff. And certainly there's the guidance document we have on that. But then there's going to be also the clinical performance. How successful are clinicians at making the decisions that they need to make? How successful are they at that? And then you have performance, and then you have making decisions, and then you have the need for truth for that as well.
Mm-hmm. It did strike me from the prior discussions about pre-analytics and scanning that there is this concrete need for evaluation essentially of that section of it, whether or not that's something that's being done by the same group that's evaluating the algorithms and their performance.
I see it as needing to sample from that space. Mm-hmm. Of pre-analytic variability, but in terms of the regulatory side, that's probably someone else's job rather than the FDA's job. And there are regulatory bodies that do concern themselves on pre-analytics. I assume I'm not totally plugged into that space, but from my standpoint, it's about sampling from that.
I mean, one from a practical standpoint, in a little bit different space than the digital pathology, algorithms are trained usually with specific signal, and sometimes it's tied to devices or tied to specific scanners if you want. So there is a differentiation between that, and in how you evaluate eventually sometimes have to do with all the pre-algorithmic parts, right? The algorithm is not going to perform equally well in everything, just because the signal is different. So you have a good point there, Alex.
And so if anyone has any questions later on, or hopefully we can, we mostly focused on the executive summary, trying to really give the scope of what this work group's about. The questions down below, I haven't given them enough thought to try and, but I imagine I want to probably wordsmith them a little bit and filter them a little bit. But in terms of getting a first draft of something to be on the Alliance website, I think we're moving forward and focusing mostly on the executive summary part.
All right, let's move on. Matthew, do you want to briefly summarize the machine learning group?
Sure. Thank you, everybody, for the discussion. Ultimately, we do have, I guess, maybe some changes to the executive summary, so I can send that later. But to summarize, we really looked at more of the perspective to, I guess, be more prescriptive on specific model operating point metrics and performance based on a specific intended use. So ultimately, models that are designed for use cases should define a clear intended use, and almost kind of designing a package insert, if you will, or a blueprint for a package insert, so that when these models these models can have defined inclusion criteria, exclusion criteria, obviously a specific intended use, being able to provide the sensitivity, specificities, et cetera, for again a given operating point. And we also discussed that the model should be able to provide some confidence or probability of their output, so that the end users are also understanding of the level of confidence that the model is providing those outputs with. We also discussed trying to define robustness across variable data sets, such that if there was some way to essentially understand or define the parameters, all the various parameters that were used with a specific training data set, and for the model to be able to potentially reject inputs or reject samples that do not occur within that intended use case, or outside the bounds of the variability of which the training set or the data set was originally trained. So essentially what we discussed was trying to be, again, more prescriptive of what a package insert or so-called package insert, or I guess best practices of being able to put a model out into the wild and actually have kind of a list of, which I guess touches on what Brandon was talking a little bit on, kind of model performance, but a little bit on kind of a breakdown and definition of the training data set and the variability that exists within that training data set, and all of the metrics of the model at a given operating point, so that the end user is more informed of exactly what the model's intended use is, and also what its intended use is not, so that they're not expecting it to do something which maybe it wasn't trained to do. We talked a lot, about some other things, but generally that's so I guess the bulk of what we were trying to kind of wrap our heads around, talking about kind of these uncertainty metrics. Again, if the model is not trained on a specific input, that it in theory should be able to reject it in some fashion, and defining acceptance criteria of a model.
Thanks a lot. I thought the rejection of sample thing is a very great idea. It reminded me sort of clinical consultations, right? You get an ID consult, and then the ID doc comes and says, hey, you should get actually a rheumatology consult, not an ID consult, right? It sounded a bit like that, right? If it's not a prostate, reject it. Or if it's a breast, if you recognize that it's a breast, you shouldn't apply the prostate algorithm. Is that kind of what you meant there?
In a sense, I mean, that would be great. So there's really no way to define this today. I think at best, we're trying to capture data that exists in the laboratory information system. But in theory, if we can learn from our lab medicine colleagues, right, they have specific reference ranges for a specific analyte or for a specific test, and they also have inclusion criteria for samples. And so, I mean, today there's a technologist that actually runs the test through the machine, but the machine and/or the human will actually or may actually reject the sample, say if the volume is too low, if the volume of that sample is too low, or say it was supposed to be received in a red top tube, but it was received in a different tube, right? So we're trying to kind of create a digital pathology version of that, where the model A has to understand all of the variability of which the training data captures, and say there's an average, I mean, if we're talking color profiles, right, or if we're talking some other variability of lab staining or variability of whatever is included in that data set, that there has to be some understanding of that, where the model essentially says it's not eligible, or it doesn't have the confidence to run on this new input because it's outside of the limits, so to speak, of what it was trained on. But
Do
that
you,
doesn't
do you
exist.
think
We're creating red top slides for machine learning. Yes, I love it.
do you think there's enough descriptive information about the data that algorithms are trained on? Because that's really what you're talking about a little bit, at least one aspect, is what data was the algorithm trained on, and whether that information is actually useful to an end user, or whether they just would ignore that and just be, take somebody's word that it gets this performance on this population given these inclusion criteria. Do we want, as a community, to really focus on being much more transparent about what are the data characteristics that went into the training of the algorithm?
Well, so Brandon, that's not really the, I get that, the importance of that, but that's not really the avenue we went down. It was actually more along the lines of, well, what are the assumptions that people are making when they're running these algorithms, right? So they're going to assume that they're going to put a slide in and get an answer out. But the intricacies of AI and ML, it's always going to give you an answer. But just like a resident, you need to know when you don't know something. And that was one of the things that we need to identify, are ways of really saying, I don't know, or this is not within the sphere of my knowledge. Those are more of the things that I think we want to focus on, and not saying what those variables are, because we don't know them. And they're going to be different for every algorithm probably. But as you develop these devices or these algorithms, make sure that one of the things that you focus on and identify are what is within the realm of possibility of me actually performing this task. Because it only, all these algorithms, they only know the space that they have seen. There is nothing on the outside. So we just have to keep that in mind.
So I'm going to push a little bit, because you're describing it, all the solutions, as coming from the algorithm. And what I'm pushing is, can we expect that the clinician, given more information about the algorithm, can be responsible for some of that?
I think that gets into the realm of decision support versus decision making, right?
But I think it's also, I think there needs to be more transparency to the end user. So say there's a model that predicts thyroid cancer, right? But there's maybe 10 different types of thyroid cancer, but this model was only trained to detect 7 of them. And the 3 super rare thyroid cancers are not included. All of the, end users ultimately will hear, oh, this model detects thyroid cancer and maybe classifies them. But the end user needs to know it can detect thyroid cancer A, B, C, and D, not E, but it can do F, G, etc., so that they have an understanding of what the very defined intended use of the model is, not just the high level use case.
Yeah, so unlike the standards, Brandon, where you're asking how well does it perform the task, we're just making sure that that task is very, very clear. Because we don't want people to overinterpret the confidence of the ability of these models.
Yeah, and at
But
the end of
aren't
the
you
day,
also wanting to get the app, for example, the algorithm or the data that you can use it also differently? Okay, it has been trained on a certain set, but you also want to take the opportunity now you have it to discover and explore more, not necessarily for clinical diagnostic decision making, but more also to get more data, like how does it work in a broader patient population. So it might not be your intended population, but you start to collect the data in such a way that you can start drawing conclusions, right? Does it work on something else as well? And then how can we validate this? And there are of course some ways that you can do it with regards to real world data collection, but also to do some extra research, right? Hypothetical testing, modeling, that sort of things. Was it also something that you discussed in your group, how to include that in this setting? And if you want to already start working on that.
So we did discuss generalizability in that sense a little bit, but maybe not as much as we discussed some of the other things. But I think we all agree that I guess performance, and not just generalizability across varying data sets, or let's call it other data sets, but also performance monitoring over time as data may change, right? There's data shifts, and sometimes there's even reclassification of things in medicine, right, over time. So so we did discuss generalizability, but also observing how the model may perform over time, where there may be changes within a given system. But all of it kind of is wrapped around this data set variability and how that data relates to at a deployment site, how their data relates to the data set that the model was originally trained on.
Carla, you wanted to make a comment as well, right?
yeah, I think that the bigger picture comment is, there's algorithms, and then there's what you take to the FDA, which is more than just an algorithm, right? There's like the user interface, there's usability, there's, what the pathologist does with whatever they're consuming from the combination of the two. And sometimes there'll be products that have more than one algorithm in there, right? So I think that very clearly specifying, what this product does, what it's for, what it's not for, and then the performance of the multiple parts of it is really important, which gets complicated, right? But then things are clear, and users won't use the product erroneously, right? Because that's a real danger here, right? This is a very new space, and you don't want users to think that the performance of the first piece is the same as the second. So I think we're going to have to be very, very clear about those things.
Great comment. Stephen, do you want to comment on what you just put in the chat? I know that chat is very good, but sometimes it's highly condensed.
Well, the only thing I was adding is that when we were talking about understanding the training data in the sphere of that space, that's not necessarily like traditional metadata like you're used to thinking about. Like, is this from a young person? Is this skin from the forehead or the back? Things like that make a difference in your diagnostic decision. But really, I've just focused on purely the image analysis algorithms, for example. It might be that tissue folds will always result in a classification X, right? Because you don't really understand your model. And you can look for these things. And so being able to rule in or out a particular image, you could just use the image. to tell you all the things that you need to know. But really, if your algorithm is a combination of image and metadata, you can't perform your test without doing that anyway. So you are going to have that metadata in that.
Other comments on machine learning? Sounds like machine learning covered a lot of different topics. All right, payers. I don't know, Laura or Esther. Laura, do you want to give it a shot?
I was, if Esther's on, she gave a really nice summary right there at the end of our working group session. She, Esther?
Sorry, I was unmute. Sorry. I was pushing all these buttons, but it didn't work. Yeah, you can go ahead if you want to do the summary.
Okay. So we discussed some of the pain points in the reimbursement space, and potential, next steps as far as how to generate evidence for supporting payment for digital pathology. So, the major discussion fell on the issue of if we're looking at digital pathology as a cost savings or increase in efficiency, that's not really sufficient information to be taking to payers, whether public or private, really, as support for this is why you need to pay for this device, or this is why you should pay more, because the payers will then reflect that as, well, great, then you should take those savings in cost or efficiency and apply those to supporting the cost of the device. But the problem is that obviously laboratories have to deal with the high upfront cost, which it can be substantial, and laboratories generally don't have that kind of money sitting around. So we talked about the other sort of major pain point being that the payment system in the United States is very disjointed. We have lots of different payers, and even within the larger Blue Cross Blue Shield and United, there are actually groups of other smaller companies that cover regions. So trying to navigate the evidence frameworks for each one of those can be difficult. So we discussed general agreement that of trying to get payment for digital pathology as a technology versus being very indication specific, that we wanted to try to look at areas for evidence generation to support the device, the technology as a whole, as opposed to sort of dividing things up into very specific indications. And so some of the short-term goals being that we could identify an optimal use case and then develop use that to develop an evidence framework. And then use the payers, so specifically Blue Cross Blue Shield makes public some of their general evidence frameworks that they use to make coverage determinations. And so trying to apply that to develop a framework for the digital pathology community in general, that potentially could be used as a starting point. And then longer term goals would be trying to develop some of the short-term endpoints for use, potentially by identifying that use case, with realizing that the longer term endpoints, and long-term follow-up of patients will still need to be done, potentially aligning those with the shorter term studies. So that's a quick summary of what we discussed.
Fantastic. Yeah, I think the paper from the payer group, which I believe was one of the first ones as the sort of formal output, is one of the top clicks. A lot of people are interested in it. It's a fantastic summary. So thanks a lot. There was also like at least 2 times when I joined, it had the largest interest. And I know that there was a lot of discussion how payers operate, right? And the outcome relevance and, you know, the technology as a whole or individual items would be great. Did you guys discuss the role of FDA authorization in relation to how payers navigate, let's call it, reimbursement decisions?
We did not get to FDA decisions within this working group, although that definitely has been a source of robust conversation in prior.
Yeah, and also if you look into, so what we did do, and thanks to Eric who joined as well, who is new. So what we did do is look into what kind of evidence are the local payers asking for. And one of the things that you do see coming up is like, has the FDA cleared the device? Of course, they also do LDTs maybe, but they also looked into what is the status of with regards to the clearance of the device. So there is some reference to it. And I think one of the things that we also identified, as Laura was saying, let's start collecting what kind of evidence are they really looking for? What do they require us to come up with? And it can be from literature studies to executing real clinical studies, longitudinal studies, whatever. But at least start to collect that information. What do they need? And based on that, as Laura was saying, have the short term and the mid and longer term value creation for digital pathology mapped out.
Other comments? All right, calling on the trainees group. Maybe as you're getting ready, the continuous learning and the practical use cases have been done sort of offline. So the trainees group will conclude the summary. I don't know who I can call on. Julia, Sarah?
I can take that. So as we're looking for trainees who are interested in regulatory sciences, we started by discussing actually our target audience, and we had come up with the 3 components of pathology residents and fellows and graduate school students and postgraduates. But we added to our list that we also want to attract early career scientists who are kind of skipping the academic or not taking the academic path, but straight going to, for example, companies. So we definitely want a very young target audience as well there. Then we talked a little bit about the practical aspects of the group and what we want to provide to the members of the trainee group, being interactive seminars, technical projects, working courses. But then we also discussed very, very short 15-minute brainstorming sessions that we could maybe host once a month or whenever they were needed. And that specifically being for people who have a very, very precise question. Let's say I'm starting this project in 2 months and I want to write an IRB and I don't know how to do that. Can you guys help me there? So really building that network of experts and members that can all contribute to the single needs that we have every day in terms of regulatory sciences. So we talked about these short brainstorm sessions, and that kind of led us to the best way how to communicate within our trainee group and how to really navigate like communication in a smart way. So we talked about Twitter and having a Twitter account, and talked about hashtags. So I think we decided on a hashtag that we want to use, which is going to be PIcc trainee group. It's kind of obvious, but I really think it makes sense. If you have other suggestions, I'm very, very happy to receive them. So we'll have a Twitter account to really give updates in a very short manner. And then talking about experts and how we can recruit more experts. And I think this really goes out to you now. I want to encourage you to take surveys, especially the expert survey, that we will post in the chat right now. It's a super short survey where you can basically choose your field of expertise and then say if you're willing to donate a little bit of your time to give maybe a session or a training, like a short course, or yeah, provide your expertise in some sort of way. That would be amazing if you could all take that. It's really short. And then if you're interested in becoming of course, a member of the trainee group, but this is really for the younger folks, I'd encourage you to take that as well. So that would be great. And you can type in your info and we can get back to you on that. So really as we're trying to build how to pitch our trainee group and raise more awareness, we're at the same time obviously looking for experts who can build this trainee group. So it would be amazing if you could take that survey. And then I want to briefly talk about our next meetings. We have scheduled a first tentative meeting for April 2nd at 3 p.m. where we want to get back together, give a couple of updates of the website, hopefully also on the Twitter account, maybe collect more thoughts and ideas on that, and really like finalize the entire, like, I don't know, slide deck, pitching package, how I should call that. And then I think the kickoff starting meeting for the trainee group would be April 23rd. That's what we discussed so far. If you have any ideas on that, or I'd be happy to take that as well. But yeah, I think so far that's what we discussed. And I'll send you the updated trainee group form. So a couple of things added, nothing really changed to the old version. We didn't cross anything off.
Julia, can I make a request for basically a kind of advertisement with this information, because I'd like to circulate it, but I don't want to compile it all. So if you can consolidate the mission and some of these links, because I can't copy and paste it actually out of the
No. Absolutely, we can circulate that in a more handy manner. Absolutely.
Because if it's just like one email that has all that, I can then forward it either FDA and also NCI. I can probably get it moving around there too.
Thank you so much. This is great. We'll definitely do that.
The survey links are going to be posted to our website page. Ula, do you have like a general idea of when the trainee page and the other pages for breakout groups are going live?
We're hoping at some point, probably within a week or so. So as soon as we get all those overview docs back from everyone today, that's really just going to help us finalize everything and then get them up sometime this week.
Okay. Thank you.
Perfect. So other comments, suggestions, project ideas, complaints? Go ahead.
Yeah, so I joined today, and I think I mentioned this in front of a couple of you probably in the training group. So I know I'm a pathologist and a hematopathologist in particular. And I know for a fact that there are a lot of trainees who are interested in this, but they don't know where to go, right? And I feel like this last session, when Julia was talking about advertising, this is critical to recruit, young minds or people who are freshly graduating, especially from the pathology, niche. So I know a lot of people who have a lot of ideas, and pathology trainees in any specialty, they sit down with experts all the time, and they hear a lot of things, and with time they come up with a lot of ideas. So this is actually like a great these people are really great source for a lot of good ideas, especially if they're taken care of right from the get-go, like from the moment that they start training throughout their graduation. So I think like that last part is very important from a pathology point of view.
Thanks. I fully agree. I mean, we have reached out through, the resident delegates. I think CAP is activated. I think this first meeting will sort of likely be a game changer, because I would say this is one of the big forces in pathology that will change the entire field. And trainees are already exposed to it, right? The board exam is already, at least in part, administered digitally. A lot of graduate and postgraduate programs are hitting pathology as well from external, right? It's not only trainees in residency and fellowships. There are computer scientists, there are a lot of industry trainees that now enter the same topic. So I think it's definitely, I believe, extremely important to have this group advocating, as well as you just outlined, create new ideas. And clearly we're struggling, right? So I think it would be phenomenal to have trainees as part of that as well.
And also, like a lot of programs have digital scanners in their departments. So there are a lot of trainees that I know as well that have a bank of slides of certain disease. So these are like great sources if you want to investigate any topic, right? And it's actually invaluable because you get somebody who collected something during their residency, and they're up to 200 slides of one single entity. So the opportunities are endless once you start to recruit those young minds into this.
Thank you.
Yeah, of course.
Sounds you're a good advocate for us.
I'm a big fan of implementing digital pathology to pathology practice as soon as possible. So I'm very happy that I stumbled upon all of this. I'm a newbie, I admit, but I have a background, and I'm happy that I found you guys, for sure.
Other comments? All right, general comments, projects, ideas, or other things you would want to throw out or want possibly some collaborators for? If not, I hand it over to Ulla. If you could maybe briefly sort of outline what's next, what we're going to do next.
Yes, just to wrap it all up here, I want to leave you guys with a couple of dates for when we're going to be meeting next. Joe, can I just share my screen quickly, please?
I thought you should, but apparently that got erased. Here you go.
Thank you. All right, so next steering committee meeting is coming up for March. That'll be on Wednesday, the 24th. And then just want to, again, draw some more attention to the dates that the trainees group mentioned for their upcoming meetings. So as it was pasted in the chat, and then all of this information, as always, will be on the website. We'll try and get the update for this meeting up as soon as possible. And we're always continuing to try and make that better. So as everyone's mentioned, just to have a really solid resource where you can find all of this information. In terms of the work group pages, our goal is to have them up within a week. So I'll just keep reiterating, if all of the leaders can just send me over their overview docs that they worked on today as soon as possible. We're going to try and get everything finalized. And that's it. Thanks everyone for joining today.
Thank you. This worked much better than I thought. I love the breakout rooms. All right, let's stop the recording.